morning to all of you. We were discussing about the chapter, the cell, the basic unit of life, and in our previous lecture, uh, we discussed about what is a cell and what is cell theory, which was proposed by such Redin and Suchman. Okay, so today we're going to discuss about type of cells. Okay, type of cell. So we categorize cells on the basis of its complexity, the type of cell. It is whether prokaryotic cell or eukaryotic cells. Eukaryotic cell generally associated with, or this cell, eukaryotic cell, they are associated with multicellular organisms. Okay, multicellular organisms like plants, animals, higher animals, etc., including human beings. Prokaryote means in unicellular, or you may call it single cell organisms like bacteria. So in this lecture we specifically talk about prokaryotic cell and its structure. When we say prokaryotic, means prokaryotic. Pro means primitive, and whenever you say K-A-R-Y, karyokinesis, or carry means we are talking about nucleus. So, living organisms have primitive nucleus. They are known as prokaryotes or single cell organisms. Okay, they have primitive nucleus. So now we will talk about, or we will study about this diagram of the prokaryotic cell. Prokaryotic cell is covered or enveloped by two layers. One is cell wall and then cell membrane. This is the outermost membrane of the prokaryotic cell. This outermost membrane of prokaryotic cell is known as, this is cell wall. Means this is outermost layer. Okay, this cell wall is made of peptidoglycans. This cell wall is made of peptidoglycans. That means sugars plus अमाइनो एसिड्स, यानी इस सेल वॉल की जो कंपोजिशन होती है वो किसकी बनी होती है? वो पेप्टिडोग्लाइकेंस की. दैट मीन्स शुगर एंड अमाइनो एसिड्स मीन्स सब यूनिट्स ऑफ प्रोटीन्स, they are present in this cell wall. When we specifically talk about this sugar unit of this peptidoglycan, and this sugar unit actually makes up the real system or this structural system of this cell wall. यानी जो स्ट्रक्चरल अरेंजमेंट होती है एक सेल वॉल की, वो स्ट्रक्चरल अरेंजमेंट जो है उसके लिए कौन रिस्पॉन्सिबल है? दैट इज शुगर. इट हैज टू बीटा लिंक्ड beta linked subunits, or you may call them structural. They are alternative structural subunits, and they are very important. इसको काफी बार पूछा गया एग्जाम्स में. NaM and NaG, and acetyl muramic acid and N-acetyl glucosamine. Okay, so N-acetyl muramic acid and N-acetyl glucosamine are the structural subunits of cell wall. They are beta-linked subunits and a part of sugar or this peptidoglycans. तो आपने इस चीज में याद रखना है कि cell wall is the outermost layer of a prokaryotic cell. It is made of peptidoglycans, sugar plus amino acids. That means, okay. So this sugar part has two beta-linked subunits which are responsible for the structural composition of this cell wall. Or the stiffness which is provided by cell wall is due to having these beta-linked subunits, that is NaM and NaG, N-acetyl muramic acid and N-acetyl glucosamine. Thereafter, the inner layer, which is cell membrane, and it is enfolded in certain areas like this. So this pink layer, this is second layer, this is our first layer, and this is second layer, which is cell membrane, or you may call it plasma membrane. Okay, and this plasma membrane is made of phospholipids plus proteins. Okay, and these enfoldings of this cell membrane, these enfoldings, हम इनकी बात कर रहे हैं, these enfoldings of cell membrane, they are known as mesosomes. Okay, and these mesosomes are thought to be the primitive mitochondria. इसको हम क्या मानते हैं? इसको हम मान के चलते हैं, it is primitive mitochondria. इसलिए क्योंकि जो उसका फंक्शन है, because of its function in respiration means ATP production and cell division, which is an additional function, okay, in addition to respiration. So because of its function, respiration and cell division, this is known as primitive mitochondria. Means after the evolution of for a so long time, this mesosome actually evolved when the cell has become eukaryotic cell, and this evolved mesosome then became mitochondria in eukaryotic cell. Because of actually we associate lysosomes with mitochondria in eukaryotic cell just because of these similar functions. Okay. So second layer is cell membrane, and then as you know that prokaryotic cell primitive nucleus, they do not have any nuclear envelope. Okay, so there is no nuclear envelope. There is no nuclear envelope, and genetic material is suspended anywhere in the cell. Okay. So inside the cell membrane, and all of you know that there is a colloidal solution which is known as cytoplasm. Why this is known as colloidal solution? Because in this cytoplasm, sugar, amino acids, electrolytes, etc., all of them are present in this cytoplasm. That's why it is known as colloidal solution. In the cytoplasm, all the major organelles are absent in prokaryotic cell. Like there is no endoplasmic reticulum, there is no Golgi apparatus, there is no mitochondria, and there is no chloroplast. Okay, so major organelles like chloroplast, mitochondria, endoplasmic reticulum, and Golgi apparatus, all of them are absent in prokaryotic cell, including there is no nuclear envelope around the genetic material. So this is genetic material, and there are some segments of genetic materials are present or suspended in the cytoplasm too. यानी यहाँ तो एक साथ जेनेटिक मटेरियल यहाँ पे आ गया, डीएनए, दैट इज द जेनेटिक मटेरियल. दिस इज genetic material, DNA, which is without any nuclear envelope. There is no nuclear envelope around this genetic material or this DNA, and when this DNA they are condensed to form chromosomes. So there is no nuclear envelope around this genetic material. So besides this genetic material, what I said that there are some segments of DNA, they are present or they are suspended in the cytoplasm. ये देखिए यहाँ पे सस्पेंडेड है, suspended segments of DNA. They are known as plasmid. So plasmid is, or they are also known as, हम इनको और भी बोलते हैं accessory genetic material. Plasmid is also known as accessory genetic material. It is suspended in the cytoplasm. Okay, and one of the most important characteristic of this plasmid that it is used in techniques of biotechnology. Okay, especially in recombinant DNA technology, where we use this plasmid or these DNA segments as vehicle DNA. ओके, यानी हमें जब कोई जीन को ट्रांसफर करना हो किसी दूसरे स्पीसीज में, किसी दूसरे ऑर्गेनिज्म में, तो हम किसकी मदद लेते हैं? इसकी. It is actually, it functions as vector, as a medium to transfer the genes from one species to another species through a biotechnological techniques. तो वहां पे इनका हम यूज करते हैं प्लाज्मिड्स का. ओके, तो हमने यहाँ पे पता किया सेल वॉल के बारे में, सब यूनिट्स, सेल मेम्ब्रेन, इनफोल्डिंग ऑफ सेल मेम्ब्रेन, this genetic material is DNA, suspended is plasmid, and there are protein factories which are also present over here. And what are the names? You know it very well. These are ribosomes. So ribosomes, as you know, they are protein factories. Ribosomes in prokaryotic cells are 70S type, means 50S plus 30S subunits. अब आपके दिमाग में ये बात आनी चाहिए कि 50 प्लस 30 80 होते हैं, यहाँ 70 क्यों लिखा? वजह, इस चीज की वजह ये होती है कि जो बड़ी सब यूनिट होती है राइबोसोम की, जैसे ये है, ये आपकी 50S है सब यूनिट, और जो आपकी ये 30S सब यूनिट, ये आपस में ज्वाइन करती है. यानी ये दोनों की दोनों फ्रीली सस्पेंडेड होती है साइटोप्लाज्म में, अलग अलग, इकट्ठी नहीं होती, अलग अलग होती है. जब ये इकट्ठी कब होगी? जब सेल को प्रोटीन्स बनाने होते हैं, तो प्रोटीन्स बनाने के दौरान ये दोनों की दोनों सब यूनिट्स एक साथ आती हैं और वो मदद करती है उस mRNA को, ताकि वो आगे प्रोटीन बना सके. ये आप पढ़ेंगे नेक्स्ट ईयर +2 में. तो एक्चुअली कुछ हिस्सा इस स्मॉल सब यूनिट का जो है वो मर्ज होता है लार्ज सब यूनिट के अंदर, तो इसलिए हम इसको काउंट करते हैं 70S type. Okay, so ribosomes are 70S type. इसमें एक बात और ध्यान करने वाली है कि यूकैरियोटिक सेल्स में there is 80S type of ribosome, but the eukaryotic mitochondria has 70S type of ribosomes. Okay, so the function is protein factory. They synthesize proteins for the cell. And there are food droplets, or you may call them oil droplets, food स्टोरेज के लिए. ओके, तो फूड स्टोरेज के लिए आपको ये चीजें मिलेगी. वन मोर थिंग very important, the presence of flagella, function locomotion, movement के लिए. तो flagella is present, is single large flagella is present, and it is made of flagellin protein. Okay, and structures like, यहाँ से आएगी, these finger-like structures which are originated from the cell membrane, they are known as pili, or you may call them sex pili or fimbriae. You may call them fimbriae, pili or sex pili. So their main function is to capture food particles and important function in reproduction, especially a very significant reproduction, which is very quite similar to that of sexual reproduction in higher organisms, is conjugation. आप मोटा मोटा जान लीजिए, जैसे कि एक बैक्टीरिया सेल ये है, एक सेल ये है, ये दोनों एक्सचेंज करते हैं अपने जेनेटिक मटेरियल को विद द हेल्प ऑफ a conjugation bridge. So this conjugation bridge is actually is formed by these sex pili or pili. Okay, so इसमें इसकी काम क्या है? To form a conjugation bridge in order to exchange genetic material between two bacterial cells of different strain. ओके, तो सीलिया की सॉरी इसकी पिलाई की बात होगी, फ्लैजिला की बात होगी, हमारे पास राइबोसोम्स की बात होगी, प्लाज्मिड, न्यूक्लियस, सॉरी प्रिमिटिव न्यूक्लियस, डीएनए, एनवेलप, और एक और चीज इसमें एक और चीज है, आउटसाइड द cell wall, so there is a gel type layer which is known as slime layer. We call it subset of glycocalyx layer, or the subset of cell wall, extension of cell wall. Okay, so its main function is it is actually shock absorber, and it provides raw material for the repair of cell wall. Okay, and it also provides protection to this bacterial cell in various chemical or physical environment. Okay, and it's subset of glycocalyx layer, this is slime layer, and it is very easy to remove this slime layer in the labs by the technique, or using technique that is centrifuge technique की मदद से आप बड़ी आसानी से इस slime layer को रिमूव कर सकते हैं in order to treat this bacterial cell. सेंट्रीफ्यूज में होता है कि उस बड़ी तेजी से घुमाते हैं उसको, जो एक्चुअली रेवोल्यूशन जो होते हैं वो काफ़ी तेज़ी से होते हैं, बहुत तेज़ी से वो चीज़ें घूमेंगी, यानी उसको बोलते हैं हम लोग RPM, रेवोल्यूशन पर मिनट, कम से कम 15 से 20 हज़ार बार वो एक मिनट के अंदर वो चीज़ें घूमेंगी, और उस वजह से क्या होगा, ये जो स्लाइम लेयर है वहाँ से वो सेपरेट हो जाएगी. So it's about the structure of prokaryotic cell. We have discussed various parts of this prokaryotic cell. Okay, so tomorrow we will discuss about eukaryotic cell, its parts and various functions. Thank you.